This lecture is on viral replication and disease. Most of the early studies that taught us how viruses replicate were on bacteriophages, or viruses that infect bacteria. So let's look at the structure of a bacteriophage. A bacteriophage has this head structure that I'm drawing, which contains its nucleic acid. And then the head region is attached to a sheath by a collar, which I just drew, and the sheath is attached to these tail fibers. The tail fibers are what makes contact with the bacterial cell and binds to a receptor on the bacterial cell. And then this whole area composed of the sheath and the head and the nucleic acid is called the capsid. And then down below we have this area called a base plate. And the base plate is a place where the virus actually will attach on to the cell so that it can insert its DNA into the cell. And let's take a look on the next slide as to how that happens. So all viruses are specific for the certain types of cells that they infect. For example, the T phages are known to infect E. coli bacteria. And here we can see a little drawing of a bacteriophage, and it will bind to specific receptors on a bacterial cell. And then there's its nucleic acid, which it will inject into the cells inside. In this particular example, we're looking at a bacteriophage infecting a bacterial cell, but viruses can infect all different types of cells, including animal and plant cells. And we'll talk about some other types of viruses that infect animal cells in the upcoming slides. When a virus infects a cell and makes more of itself, we say it's in the lytic cycle. Oops, I forgot an L in that cycle. C-Y-C-L-E. And in the lytic cycle, the virus first invades the cell, and then it makes more of itself, packages the viral particles, and then it destroys the host cell when the cell bursts open, releasing the new viral particles. And we say that this virus is virulent or has virulence. And that means that when a lytic virus causes a disease, it does so by destroying the host cell. And then this slide shows the steps of the lytic cycle. And so first we have a bacterial cell in this case, and drawing a little bacteriophage that just recognized its cell and is sitting on the cell. Its tail fibers recognize its host cell, and then it's going to bind with its base plate to the host cell and inject its nucleic acid into the host cell. And that DNA will then replicate and make many copies of itself, and it will also make protein, so make messenger RNA, which is then used to make protein, and then those proteins build the new viral particles. That's called assembly. And so just building some heads here and some tail fibers, nucleic acid into the new heads. So we have a whole new population, and understand there would be millions of new viral particles produced, and then they burst out of the cell, destroying the cell, and these new viral particles can then go on to infect new cells. And then some viruses also have a lysogenic cycle, and what that means is that the virus can house itself in the host cell for a long time. And these viruses are called temperate viruses. So a temperate virus is a virus that is replicating along with the host cell by what is called lysogeny. Okay, so it inserts itself into the host genome, or the host DNA, and then basically stays there until the host replicates. A prophage is a virus that has inserted its DNA into the host DNA and has basically become part of the host DNA, shown in this picture here. And then when the host cell divides, it replicates the viral DNA along with the host cell's DNA. Let's look at a virus that's more relevant to us, HIV, or human immunodeficiency virus. And it's called human immunodeficiency virus because it makes us deficient in our immune system. Okay, this is the virus that causes AIDS, or acquired immunodeficiency syndrome. And so the difference between HIV and AIDS is the symptoms of what you get from being infected by HIV. And so you get the symptoms of being immunodeficient. And so it affects T cells of the immune system. Let's look at the steps involved in HIV infection. So HIV is surrounded by a membrane. It's an RNA-containing virus. It recognizes the T cell, fuses with the T cell, and inserts its RNA into the cell. And then that RNA is used as a template to make DNA by the enzyme reverse transcriptase, which is actually brought in by the virus. And then the DNA that is formed from the RNA goes into the cell's nucleus, where it recombines with the host genome. And then the viral DNA can both replicate, and it can be also used to make viral RNA, viral messenger RNA, which goes out of the nucleus into the cytoplasm to produce new viral particles. So proteins are produced basically that are assembled, and then they go out of the cell in a process called budding. And budding eventually lyses the cell and destroys the cell. So we could put these numbered steps in. 1, 2, 3 is the RNA is inside the cell. And then in 4, DNA is formed. It goes into the nucleus. It recombines with the host DNA. Step 6, then replicates or makes messenger RNA, or both. New viral proteins are produced, and they assemble into viral particles. The new viral particles bud from the cell. Approach, the HIV virus can also insert itself into the host genome and can stay there as a provirus. And as a provirus, it is in the lysogenic phase, where it is just part of the cell's genome. And so let's talk about viral evolution. So viruses are actually thought to have come from cells. And so the DNA of cells became viruses and went out of the cell and became its own entity, perhaps through mutation, and then traveled from one cell to another cell, and further mutation happened so that the viral particles were able to evade the immune system. And as we know, viruses replicate very quickly, and they also can mutate and evolve very quickly. I mean, just think about what we know about the flu virus and how often people need to have a new vaccination because the flu has mutated once again, allowing it to evade our immune systems. Let's talk about some other infectious diseases caused by viruses. For example, rabies virus, which is passed on through animal bites, or chickenpox, which is passed on by skin contact, but can also be airborne. And it can persist as a provirus for many years in nerve cells. In fact, it can come back in adults and then present itself in a new way, in which case it's called shingles. And with shingles, people get skin rashes, feverish, and also pneumonia. It can be very painful. And so those of us who had the chicken pox a long time ago, I myself am one, have gone out and gotten the shingles vaccine so that we don't get this virus coming back through our nerve cells and infecting us once again. And then let's look at the prevention and treatment of viruses. And so for one thing, you have antiviral agents or antiviral drugs. Drugs like acyclovir, which is used against herpes simplex virus and also against chickenpox, and azodothymidine, which inhibits the reverse transcriptase of HIV, and also protease inhibitors, which inhibit viruses like HIV. And various combinations of these drugs are then used to prevent HIV infection or to slow the replication of HIV after it's already infected a person. But the greatest development in getting rid of viruses has been through vaccination. And so for a vaccination, a virus is inactivated, usually by boiling, and then the weakened or inactivated virus is injected into a person to stimulate the immune response. And sometimes a virus will be attenuated, which means that it's been genetically altered so that it can't possibly infect a person. And then it can be used as a vaccine. It's a more trustworthy vaccine. But often people need a booster shot, which is an additional dose of the vaccine sometime later, just to, just what it says, to boost the immunity of the person. Another strange thing about viruses is that some of them cause cancer. For example, HPV, or human papillomavirus, which is transmitted by sexual contact, causes cervical cancer. And fortunately now there is a vaccine that both men and women can get to prevent cervical cancer. And then also there's Epstein-Barr virus, also transmitted through physical contact, which leads to Burkitt's lymphoma, or cancer of the lymph system. There is human T lymphotropic virus, which is transmitted in multiple ways that leads to leukemia. And also hepatitis B virus, which is transmitted through body fluids and sexual contact, can lead to liver cancer. And so all of these cases involve the virus entering the cell and then going into the lysogenic phase. But what's happening is it's usually inserting itself into regulatory genes of the cell cycle and mutating those genes. And so there are many genes that are involved in controlling the timing of mitosis. And if a virus is creating a mutation in that, that can lead to uncontrolled mitosis. And then if a cell keeps dividing, it can then become a tumor, which eventually becomes a cancer. And so that ends our lecture on viral replication and disease.